So what is glycolysis? It is simply the splitting of a glucose molecule into two pyruvates. Simple, right? Well, the overall process requires 10 steps of enzyme-catalyzed reactions, and our goal today is to ensure that you understand the chain of reactants and products which are linked together in the process called glycolysis. In other words, how and why glucose is converted into two pyruvate molecules. Before dividing into each step, why is this biological process so important? Every cell in your body uses ATP, adenylyl triphosphate, as an energy currency to regulate proteins and catabolic pathways, transport substances across membranes, provide energy for mechanical work, etc. Perhaps the simplest way to think of ATP is to consider the energy currency of cells. If work needs to be done, ATP consumption will almost always be involved. Glycolysis generates a little ATP itself, but more importantly it creates two pyruvate molecules, which are critical in aerobic respiration, which goes on to create 36 ATP. Now, on to the actual pathway. There are two major pathways in the step, a preparatory phase, shown on the left, and a payoff phase, shown on the right. In the preparatory phase, two ATP are consumed, and the glucose molecule is split into two identical molecules of glyceraldehyde 3-phosphate, G3P. G3P is subsequently converted into pyruvate via the last 5 enzyme catalyzed steps. This is the payoff phase. By the time pyruvate is created, there is a net gain of 2 ATP. Let's look at each step in a little more detail. Remember, the outcome of the preparatory phase is to create two identical glyceraldehyde 3-phosphate molecules. This means that we will add 2 phosphates during the preparatory phase, since each G3P needs a phosphate, and glucose and has none to begin.
Now let's begin our exploration of the individual steps, beginning with the addition of the first phosphate molecule to glucose in step 1. The first phosphate is added to the most open oxygen with the assistance of hexokinase. This is a priming step during which energy is invested in the glucose molecule by breaking down ATP into ADP and a phosphate group, which is added to the glucose molecule. Next, the glucose ring is isomerized into fructose by phosphohexose isomerase. Because it is an isomerization, nothing is added or removed, just rearranged. Why might our cells put the energy into this isomerization? This question will be addressed in step 4. In step 3, another ATP is invested into the glucose molecule with the assistance of phosphofructokinase 1. The result is that a second phosphate is added, once again to the most open oxygen. This step, like step 1, is irreversible. Its delta G is highly negative because the breakdown of ATP is highly favorable energetically. One important fact about step 3 is that it is the first committed step of glycolysis. The products of step 1 and 2 are used in other metabolic pathways. As you can see, fructose 1,6-biphosphate is beginning to look symmetrical and is ready to be split in half by outerlase. Outerlase splits the linear form of glucose by performing a carbon-carbon cleavage between the alpha and beta carbons of the ketone. If the glucose had not been isomerized in step 2, this step would lead to a carbon-carbon cleavage which would result in a 2-carbon molecule and a 4-carbon molecule.
From step 4 we have two 3-carbon molecules, glyceraldehyde 3-phosphate and dihydroxyacetone phosphate. The two are so similar that triose phosphate isomerase is able to isomerize one into the other in this slightly endergonic step. In step 6, G3P is oxidized to 1,3-bisphosphoglycerate by G3P dehydrogenase, while also reducing NAD+ to NADH and releasing a proton. This step depletes G3P levels, and if you remember back to step 5, The isomerization is based on an equilibrium between G3P and dihydroxyacetone phosphates. Since G3P is depleted, dihydroxyacetone phosphate replaces it, which can then be exhausted in this step. This dehydrogenation is slightly anergonic, but thanks to the next step is able to proceed. For step 7, a phosphoryl group of 1,3-bisphosphoglycerate is transferred by the enzyme phosphoglycerate kinase to yield an ATP and a 3-phosphoglycerate. The step's highly exergonic and is actually why steps 5 and 6 are able to proceed. This leaves us at this point with having used 2 ATP molecules and having resynthesized 2 ATP molecules, as well as having reduced 2 NAD pluses to NADHs. Step 8 is a two-part step characterized by the enzyme phosphoglycerate mutase exchanging a phosphoryl group with the proton group on C2, then exchanging this proton from C2 to the phosphoryl group on C3, resulting in a 3,2 phosphoryl shift and a regenerated phosphoglycerate mutase. This all sounds intimidating, but what's going on is simply an isomerization that places the phosphate group in a position where its bond energy can more easily be captured.
Next, enolase promotes a reversible removal of a water molecule to form phosphoenolpyruvate. This dehydration creates a very important double bond which is used in the final step of glycolysis. In this final payoff step, pyruvate kinase transfers the phosphoryl group from phosphoenolpyruvate to ADP, resulting in the creation of another ATP molecule and an enolate. This enolate tautomerizes into what is commonly called pyruvate. We now have 2 identical pyruvate molecules, each containing 3 carbons from the original glucose sugar. What did it cost us? A simplified chemical equation for glycolysis is below. We converted 2 ADP into 2 ATP, which is a benefit as ATP is an energy currency used by many reactions. We also separated the 6 carbons of glucose into a more usable 3-carbon molecule called pyruvate. The water and hydrogen byproduct are negligible in living systems, however the NADH product is noteworthy. NADH is later reduced by the citric acid cycle to create ATP at a rate of 2.5 ATP per NADH. Glycolysis stops with the formation of pyruvate. Pyruvate goes on to be converted into a couple of different products. In humans, it is converted into acetyl-CoA under aerobic conditions and lactic acid under anaerobic conditions. Other species, such as bacteria, convert it into ethanol. These alternative fates of pyruvate represent different organisms' ways of getting the most energy out of a single, original glucose molecule.